Hello everyone, in this video we are going to study about hemostasis. First of all, before moving on to the topic, one thing that we have to note is hemostasis and homeostasis both are different terms. This hemostasis is not homeostasis. It's not homeostasis. So what is hemostasis? The answer resides in the word itself. Hemo means blood, stasis means to stop. So we are going to stop the loss of the blood. So preventing the loss of the blood with the help of the formation of clot is known as the process of hemostasis. So there are a sequence of events through which this process occurs. So first of all, vasoconstriction occurs, and this is the first event of hemostasis. So how vasoconstriction occurs? It could be due to the nervous reflexes, or we could say the neurogenic reflexes, which are caused by the pain or trauma caused to the blood vessel. Local secretion of the factors like endothelin. Endothelin is a potent vasoconstrictor. And then the transient effect. It means this vasoconstriction is a transient effect. It means after some time it will subside. Like it will be there for few minutes and it will subside. And this vasoconstriction occurs due to the myogenic contraction, means the contraction of the smooth muscles present in the walls of our vascular tissues, vessels. Second process that is leading to hemostasis is platelet plug formation. That is a very important process and occurs in two phases. First is the primary platelet plug formation or the temporary platelet plug formation. So primary hemostasis, how it occurs? So whenever there is an injury, whenever there is a damage to our vessel, as we know that vessel walls are lined by endothelial cells. Endothelial cells. And when this vessel wall gets damaged, this endothelium gets removed from here and it will expose some subendothelial components. It will expose subendothelial components, and these subendothelial components are von Willebrand factor and collagen that are going to promote the adherence and activation of the platelets that will lead to formation of a platelet plug. So how these von Willebrand factor and collagen helps in adherence and activation, we'll study about it later, that there are certain receptors present on the surface of platelets which are going to bind with these subendothelial components, von Willebrand factor and collagen. Now how does platelet activation takes place? That we know that platelet activation occurs at the time of primary hemostasis. So how this activation takes place? So platelets are generally small rounded disc shaped cells. They are going to transform their shape from small rounded disc shaped cell to flat plates with spiky protrusions, and these spiky protrusions are known as philopodia. They are basically like pseudopodia, false feet. They are also known as philopodia. So these spiky protrusions increases the surface area of the platelet cell by several folds. So three processes take place while platelet activation: adhesion, aggregation and secretion. What happens in adhesion? This platelet is going to adhere itself to the endothelium. It is not attached to endothelium directly. It will take place with the help of this von Willebrand factor. Von Willebrand factor will help in the adhesion of the platelet, and collagen also helps. Second is the aggregation. Aggregation helps. Aggregation process occurs with the help of fibrinogen. This is the receptor. This is the receptor present on the surface of platelet. Glycoprotein second B, glycoprotein second B third A. This is a receptor present on the surface of the platelet. Thrombin, ADP, platelet activation factor all helps in the aggregation. Third is the secretions. In the secretion process, the platelet, after getting activated, releases its granules. There are two types of granules present in platelets: alpha granules and delta granules. Delta are basically also known as dense granules, and this granule release occurs with the help of or triggers or this ADP and thromboxane A to promote this granule release. Now this is the activated platelet. You can see there are several receptors present on the surface of platelets. These are the receptors present, glycoprotein receptors or the G protein coupled receptors. And there are certain other things that are getting released, like von Willebrand factor, fibronectin, platelet derived growth factor, factor 5th and 8th, which are the factors for coagulation. Fibrinogen, thrombospondin, thromboxane A2, heparinase, ADP, platelet factor 4, thrombin, and they are helping in from platelet addition to moving on to platelet aggregation. Now second process that occurs, that is the secondary hemostasis, which is basically the formation of definitive hemostatic plug or the permanent hemostatic plug or platelet plug, definitive platelet plug. So what happens in the secondary hemostasis? The platelets that have aggregated here, there will be deposition of fibrin over them. So in this diagram we can see how fibrin is deposited over them. Well, how this deposition of fibrin occurs? So first of all, there is some vascular injury, there is some trauma to the vessel wall, which lead to the release of the tissue factor. What is tissue factor? Tissue factor is nothing but a procoagulant glycoprotein. What is procoagulant? Means that is not having any coagulative property but have a potential to be a coagulant. So it is a procoagulant glycoprotein. Basically are smooth muscles or fibroblast cells, which bind with the factor 7th and activates it via the cascade. So we will study about this coagulation cascade later in this video also. It leads to generation of the thrombin. This thrombin will activate the fibrinogen to fibrin. Okay, so this is the process that we'll study about in detail. Now this is clot stabilization and clot retraction. The one question that is generally asked is, what is clot retraction time? So the time required by the platelet plug to stabilize or to settle down or to retract is known as clot retraction time. So clot retraction time is basically 20 to 60 minutes. And if this is not happening, if clot retraction is not happening, it denotes or it implies that the platelet count of the blood is very low. So that's why clot retraction is not happening, because platelet helps in the clot retraction. So how this happens? Like, this is the platelet having this false feet, filopodia. Fibrins are getting deposited or getting entangled in this filopodia, and when the platelet contracts, it leads to internalization of the fibrin. It gets cemented between the platelets and stabilizes the clot. So it leads to consolidation of the clot, and which results in clot retraction along with the contraction of platelet. So there's a formation of a solid and a permanent plug that is accelerated. The Vihos formation is accelerated with the help of thrombus and calcium. So we know that thrombus and calcium are going to accelerate or favor the process of formation of the permanent plug or the clot retraction. So if there are no platelets, there will be no clot retraction. Next, before moving on to the coagulation cascade, let's have a look on the platelets. So what are platelets? Platelets are nothing but a disc-shaped enucleated cells. So enucleated means they lack nucleus. They do not have nucleus. Their shape is like a disc, and they are formed from the megakaryocytes. They are cell fragments which are shed from the megakaryocytes in bone marrow into the bloodstream. This is the mini platelet that I have drawn over here. These are several receptors present in the membrane of the platelets, like this: glycoprotein first B, glycoprotein 1A second A, glycoprotein second B third A. So these are the different types of receptors present on the membrane of platelets. This is a G protein coupled receptor. G protein coupled receptor is usually represented like this, with seven molecules. One side is inside the cytoplasm and one end is outside, externalized. So this is our G protein coupled receptor. So this G protein coupled receptor is a variety of receptors. So they are G protein coupled receptors are type basically, and these are the examples of G protein coupled receptor, like PAR, protease activating release receptor, protease activated receptor 1. So this PAR1 will bind to different things, like it will bind to thrombin. Now ADP will bind to this. This is also a G protein coupled receptor. Example of G protein coupled receptor: P2Y1 P2Y12. These are all G protein coupled receptor present on the surface of this platelet. Inside the platelet there are granules. There are two types of granules that we have discussed earlier, alpha granules and delta granules, also known as dense granules. Alpha granules contain fibrinogen, clotting factor 5th, von Willebrand factor, fibronectin, platelet factor 4, platelet derived growth factor, transforming growth factor beta. These fibronectin and all these factors are going to help in the wound healing, and these three factors will help in the coagulation process. Now dense granules contain ADP, ATP, calcium, serotonin and epinephrine. So now this thrombin will activate the platelet via binding with the PAR, that is protease activated receptor 1, and P2Y1, P2Y12 ADP binds to these receptors. Now this is a very kind of detailed diagram of how these receptors work. So this is the GP yani ki glycoprotein second A third B receptor, which is going to bind with the fibrinogen and going to help in the aggregation of the platelets. This GP first B will bind to the von Willebrand factor, and GP first B bind to this von Willebrand factor, and there's another factor that we studied over here was the GP1A. So GP1A is not depicted over here. Well, it binds with the collagen present in the subendothelial space. If there is a deficiency of this GP second A third B factor, it will lead to this syndrome, Glanzmann thrombasthenia. And if there is a deficiency of glycoprotein first B receptor, it will lead to a syndrome, Bernard-Soulier. If there is deficiency, congenital deficiency of von Willebrand factor, it will lead to von Willebrand disease. So these three deficient syndromes or diseases are congenital or hereditary diseases. Now the most important topic, coagulation cascade. Coagulation basically takes place from two pathways. There's an intrinsic pathway and there's an extrinsic pathway. This is your intrinsic pathway, this is your extrinsic pathway. So what are the meaning of this intrinsic and extrinsic pathway? So extrinsic pathway, when does this pathway activate? So whenever there is a trauma to the vessel wall, or there is a trauma to the tissue, there is a damage to the tissue, it will lead to release of the tissue factor, tissue thromboplastin. There are several tissue factors, there are variety of tissue factors. So tissue factors gets released like tissue thromboplastin, which will lead to activation of the 7th coagulation factor. First of all, let's have a look on these clotting factors. So there are several clotting factors over here, like fibrinogen is our factor 1. Prothrombin factor 2, tissue factor, that thromboplastin is factor 3rd, calcium is itself a factor 4. So these all are the name of the factors present in your textbooks already. The factors that we have to remember, basically the most important factors are this: anti-hemophilic factor, because if this factor is not present, coagulation does not take place. It leads to bleeding disorder, hemophilia, if this factor is absent. So this is known as anti-hemophilic factor. And there is this factor 10, that is the process from where intrinsic and extrinsic pathway collide. They merge together. The process after this factor will remain same for both the pathways. So this factor is known as Stewart factor or Stewart-Prower factor, factor number 10. Factor 12 is Hageman factor, from where the process of intrinsic pathway coagulation starts. And calcium, which is a mineral that itself a factor, it is factor 4. So let's have a look on this extrinsic pathway. So tissue factor is released, tissue factor activated this A denotes the activation. Activation means now this factor is able to do the proteolysis. It can break, it can cleave the proteins. So it gets activated to 7th A factor. That is basically our serum prothrombin conversion accelerator or proconvertin, a stable factor. So this PL over here is platelet phospholipid, and TPL is the tissue thromboplastin. So these will help in the activation of this 10th factor. Then activated 10th factor will lead to formation of the thrombin from the prothrombin. Again, this 5th A factor is going to contribute in this process. Then thrombin, with the help of calcium, converts fibrinogen into fibrin. Fibrin gets converted to stabilized fibrin polymer. Polymerization of fibrin occurs, that leads to long chains of fibrin that will act as or help in the formation of the permanent permanent plug. Now this is the intrinsic pathway. So how does this intrinsic pathway get activated? Like we have already discussed that extrinsic pathway is activated through the tissue damage or the vascular injury. Now here in this intrinsic pathway, after vascular injury, not from the release of tissue factor, the pathway gets activated because of the contact between the exposed collagen and the platelets, and these factors, the factors present in the blood, gets exposed to collagen, negative surface charge, and HMW kininogen. What is this HMW kininogen? It is high molecular weight kininogen or kallikrein. These are the factors who are going to promote this process of intrinsic pathway coagulation. So this 12th-A will get converted to activated 12-A. Now this high molecular weight kininogen will come and convert this help in the conversion of the 9th factor to activated 11th factor to activated 11th factor, then 9th to activated 9th factor, then again phospholipids, calcium, 8th factor, activated 8th factor is going to help in the formation of the activated Stewart-Prower factor. So how to remember this? We can remember it like this: 12, 11, 9, 10. 12, 11, 9, 10. This is 12, 11, 9, 10. It's like a seedhi ginti, and then after this like ulti ginti, and then after this seedhi ginti. 12, 11, 9, 10. And between 9 and 10 there will be another factor, 8. 8, 9, 10. So 8 is the factor that is going to help in the conversion of the 10th to activated 10th factor. And this is very easy, 7 and 10 only. Well, tissue thromboplastin itself a factor 3rd. So stabilized fibrin clot. So this is the process of coagulation via intrinsic pathway and extrinsic pathway. So we have studied the difference between intrinsic and extrinsic pathway. Extrinsic pathway occurs by tissue injury, tissue damage, vascular injury, which lead to release of the tissue factor, that is 3rd factor. So it starts with the 3rd factor, and here intrinsic pathway starts because of the contact with the exposed collagen and starts with the 12th factor. So that's the end of the video. In this video we studied about hemostasis, the 3 phases of hemostasis like vasoconstriction, platelet plug formation, and the clot retraction. We studied a bit about platelets and the receptors present on its cells, on its surface, and then we studied about the coagulation cascade. The most important part of the whole video is coagulation cascade, which is often asked in the exams. And after coagulation cascade, there is a process of anti-clotting mechanism. Anti-clotting mechanism that we'll discuss in the next video. Anti-clotting mechanism is not opposite of coagulation cascade, okay? It is not, you're not going to write it in a reverse direction. There's another whole different process. Tissue plasminogen activators, they are different process. So we are going to discuss it in a very short video. Okay, thank you.